So moving to the issue of mismatch repair testing in pancreatic adenocarcinoma, we know there are a variety of ways to do it. Classic ways are using IHC to look for loss for protein expression, or for PCR analysis looking for microsatellites. Increasingly, and I would say in particular in pancreatic adenocarcinoma, because of some of the advantages that next generation sequencing brings in terms of tissue conservation and learning about other therapeutic opportunities, we have ways of assessing mutation load and bioinformatics analysis to evaluate MSI sensor score. Here's a reminder regarding pembrolizumab approval, and these are the data that supported that approval. Remember, these were non-randomized studies, but there were 6 patients with pancreatic adenocarcinoma in the 5 studies, and there was a high response rate identified in this particular population. And our group chose to evaluate this in some detail and confirm that mismatch repair deficient pancreatic adenocarcinoma is rare. We saw it in about 1% of patients. In fact, in this analysis of a relatively large cohort, all of these patients had an underlying Lynch syndrome, so they had a germline mutation in associated mismatch repair deficiency gene. We saw that these patients had loss of protein expression, high mutational load, and an elevated MSI sensor score. And looking at this graphically, you'll see the typical individual pancreatic adenocarcinoma has 3 to 4 mutations per megabase, but those with mismatch repair deficiency, and again, in this analysis, all germline Lynch, had a high mutational load. And there's a group of individuals here which have a more intermediate mutational load, for which we're currently learning whether or not this might be a possible subgroup for which immune-based strategies may have a role.